are now looking at question number 18 from the GATE Biotechnology 2023 paper. The question is, which one of the following is required for the development of B cells in the bone marrow? Option A, stromal cells. Option B, dendritic cells. Option C, Kupffer cells. And option D, natural killer cells. So to understand this, let's learn about B cells and their development process in the bone marrow. B cells or B lymphocytes that derive its name from the bursa fabricius of birds is a key player of the adaptive immune system that is responsible for humoral immunity in mammals. So B cell production in humans is a lifelong process that starts in the fetal liver intrauterine and in the bone marrow after birth. So the mature B cells can have 1 to 1.5 into 10 power 5 immunoglobulin receptors for the antigen. So the B cells also interact with T helper cells and then they proliferate and switch the isotype, that is, they switch the class of immunoglobulin that is produced while retaining the same antigens, and this determines the binding specificity to the antigen. So the B cells develop from hematopoietic stem cells and they attack invaders that are outside the cells, that is, they attack any foreign antigens. Let's look more into the details of B cell generation. So in adults, B cell development occurs in the bone marrow and does not require contact with any antigen. So during this time of B cell generation, these B cells rearrange the genes for their heavy and light chains and synthesize the cell surface immunoglobulin M, IgM, which acts as their initial antigen receptor, that is the B cell receptor BCR. So the BCR complex includes membrane-bound immunoglobulin, that is small m, IgM, and the signaling chains CD79A and CD79B. So the immature transitional B cells exit the bone marrow and enter the periphery, where they further mature in the secondary lymphoid organs like spleen. So if these cells do not encounter antigen, they will die soon within a few weeks by apoptosis. On the other hand, if these mature B cells encounter specific antigens, they undergo activation, proliferation, and differentiation, leading to the generation of plasma cells and memory B cells. The B cell development and maturation from the stem cells in the bone marrow occurs in several stages. The earliest stage is the pro-B cell and then pre-B cell, which then becomes an immature B cell, which later matures to form the final mature B cell. So let's look further into the details of how each of these develop. So firstly, there's the generation of pro-B cells. The earliest stage of antigen-independent B cell development is the progenitor B cell, which is abbreviated as pro-B cell stage. So here, pro-B cells can be divided into three groups based on their expression of TDT and CD45R. The early pro-B cells express only the TDT, and the intermediate pro-B cells express both TDT and CD45R. The late pro-B cells express CD45R and have low expression or the downregulation of TDT. Now, what are these? So CD45R is a receptor for cell growth and differentiation. It remains expressed on the surface throughout the remainder of B cell ontogeny. And there's another receptor, CD43, which regulates adhesion. And then TDT is a terminal deoxynucleotide transferase. It's an enzyme that contributes to the variation of antigen receptors by the random addition of nucleotides to the single-stranded DNA at the junction of recombination of the immunoglobulin heavy chain genes during the B and T cell development. So as the cells progress through the pro-B cell stage, they rearrange their immunoglobulin heavy chain genes and begin to express CD43, CD19, and so CD19 is a co-receptor which works together with CD21 and CD81, and then RAG, recombination activating gene, RAG1 and RAG2. So as the late pro-B cells pass into the pre-B cell stage, they downregulate TDT, RAG1, RAG2, and CD43. So all these are downregulated when the pro-B cells later transition to become pre-B cells. So the pro B cells also express c-KIT, which binds to stem cell factor expressed on bone marrow stromal cells. So this induces the pro B cells to proliferate and differentiate into precursor B cells, which are the pre B cells. Now the pro B cells, the progenitor B cells, have become pre B cells. So these pre B cells can be divided into large mitotically active pre B cells and small non-dividing pre B cells. So both these large and small pre B cells express the Ig mu heavy chains in the cytoplasm and the pre B cell receptor complex on the surface. So the large pre B cells have successfully rearranged their immunoglobulin heavy chains. So as these cells pass from the large pre-B cell group into the small pre-B cell group, they begin to rearrange their immunoglobulin light chain genes and upregulate RAG1 and RAG2. So the pre-B cells, they express IL-7R and are stimulated to divide and differentiate using IL-7R. So IL-7 is a hematopoietic growth factor secreted by bone marrow stromal cells. So this helps drive the growth and differentiation of lymphoid progenitor cell lineages. We have so far learned about pro and pre-B cells. The next stage is the immature B cells. So the final stage of B cell development in the bone marrow is the immature B cell stage. The immature B cells have successfully rearranged their light chain genes and they expressed IgM. So again, the RAG1 and RAG2 expression is downregulated. So as the immature B cells develop further into mature B cells, they begin to express both IgM and IgD on their surface. So these mature B cells then exit the bone marrow and migrate into the periphery. So after exiting the bone marrow, B cells go through a phase known as the transition phase before they form mature B cells in secondary organs like spleen. So here's a pictorial representation. The transitional B cells pass through two transitional stages, T1 and T2. So throughout their migration from the bone marrow into the spleen and after the spleen entry, they are considered T1 B cells. So within the spleen, the T1 B cells transition into the T2 B cells. So the immature B cells are very sensitive to antigen binding. So if they bind to self antigen in the bone marrow, they die. So the B cells that do not bind to self antigen express D chain and the membrane IgD and IgM about the time they leave the bone marrow, and they become mature naive or resting B cells. So if these mature B cells encounter specific antigen, they undergo activation, proliferation and differentiation, leading to the generation of plasma cells and memory B cells. So this is the whole process of B cell maturation and development. So 1 to 3% of all the B cells produced in a day would reach the recirculating pool. In the previous slide, we just saw that the B cells that react or bind to their self antigens within the bone marrow, they die. So this is the negative selection of self-reactive B cells. So the negative selection of many self-reactive B cells occurs in the bone marrow. So it's also called clonal deletion. So this limits the development of antibody-mediated autoimmunity. So if the immature B cells that express membrane-bound IgM recognize the self-antigen, some of the immature B cells undergo apoptosis. Some of them undergo editing of light chain genes to produce a different light chain that, when combined with a heavy chain, does not recognize the self-antigen. So the negative selection of some of the self-reactive B cells occurs in the periphery. And not all these self-reactive B cells are eliminated in the bone marrow because not every self-antigen is expressed in the bone marrow. So but still this mechanism is under investigation. It's not fully understood. So in the whole process, there are several cytokines and transcription factors that are involved in the B cell development. So several cytokines affect B cell development. So common lymphoid progenitors, CLP, are responsive to IL-7, interleukin 7, which promotes B cell lineage development. The mice that are deficient of IL-7 or the IL-7 receptor exhibit an early arrest in B cell development at the pro-B cell stage, which is the first stage of B cell development. And another cytokine is Bliss. So B lymphocyte stimulator signaling through its receptor BR3 is important for the survival of premature B cell stages from transition stage onwards. And then IL-4, IL-3 and low molecular weight B cell growth factor LBCGF are also important in initiating the process of B cell differentiation. There are also several transcription factors in B cell development. So, B cell generation is regulated by multiple transcription factors, including E2A and EBF, which coordinately activate B cell genes. And PAX5 ensures B cell development to B cell lineages by restricting transcription of lineage inappropriate genes and by activating expression of B lineage signaling molecules. And then SOX4 and LEF1 promote the survival and proliferation of B cells. And then IRF4 and IRF8, which terminate pre-BCR signaling by IRF4 and promote differentiation to small pre-B cells. And then BCL6 expression is required for germinal B cell differentiation and generation of memory B cells. So, in contrast, expression of B-lymph1 suppresses BCL6 and is required for development of immunoglobulin secreting cells and maintenance of long-lived plasma cells. Further, stromal cells are an important component in the whole process of B cell development. So, we just learned in the pro B cell development, we saw that they express c-KIT, which binds to stem cell factors expressed on the stromal cells in the bone marrow. So, this also induces the pro B cells to proliferate and differentiate into pre B cells. So, all these are the stromal cells. So, the term stromal cells refers to highly heterogeneous class of connective tissue cells that build the infrastructure of any organ and fulfill a variety of fundamental role in health and disease. They often regulate immune responses in the tumor microenvironment as well. So, in context with the B cell development, stromal cells also retain the developing cells, B cells, in the bone marrow. They also make CXCL12, which binds to CXCR4. So, they produce cytokines that guide the development of B cells. So, the hematopoietic stem cells start at the osteoblast and endothelial cells. So, the pre-pro B cells then need CXCL12 and which gives signal to move to the pro B cell stage. The pro B cell also needs IL-7. So stromal cells produce and release IL-7. So therefore they play an important role in the regulation of B cell development in the bone marrow, mainly by controlling the size of the B cell compartment. So in the bone marrow, different types of hematopoietic stem cells also develop. So recalling our given question, we had other options as well. So of the other options, we had Kupffer cells. So these are specialized macrophages located in the liver. So their activation is responsible for early ethanol-induced liver injury, which is common in chronic alcoholics. So these Kupffer cells release factors that activate hepatic stellate cells, that is HSCs, that lead to the disease. Further, there are dendritic cells or the Langerhans cells. So these are antigen presenting cells. So the dendritic cell acquired its name because it is covered with long membrane extensions that resemble the dendrites of nerve cells. So there are 4 types of dendritic cells. So there are Langerhans cells, interstitial dendritic cells, myeloid cells, and lymphoid dendritic cells. So the dendritic cells also arise from the hematopoietic stem cells via different pathways and in different locations. So the dendritic cells again acquire the antigen by phagocytosis. And another type of dendritic cell, the follicular dendritic cell, they do not arise in the bone marrow and they have a different function from the antigen presenting dendritic cells. The other group of cells forming part of the lymphoid lineage are the natural killer or the NK cells. So the NK cells are large granular lymphocytes that kill the tumor cells and virus infected targets. So these cells constitute 5 to 10% of the lymphocytes in the human peripheral blood. So they do not have membrane receptors for antigen like the T cell and the B cells, but they have NK cell receptors. So these NK cells adhere to infected target cells and induce their cell death through delivery of apoptotic signals by programmed cell death in multicellular organisms. And this is mediated by perforins, which are pore forming cytolytic proteins, and by granzymes like serine proteases released by cytoplasmic granules within the cytotoxic T cells, and tumor necrosis factors, that is cell signaling proteins involved in systemic inflammation, and these are types of cytokines. So Fas ligand is a type 2 transmembrane protein expressed on cytotoxic T lymphocytes and natural killer cells. So it binds with the Fas receptor on target cells, which induces programmed cell death in the Fas-R carrying target cell. And these NK cells can also kill through antibody dependent cell mediated cytotoxicity mechanisms via cells surface receptors for constant domains present on immunoglobulins. Now, coming back to a question, which one of the following is required for the development of B cells in the bone marrow? So, like we saw, option A, stromal cells, is involved in the development in the bone marrow by releasing several cytokines. So, the other cells are dendritic cells and NK cells are immune cells, while the Kupffer cells are involved in liver injury. So, that answers our question. So, stromal cells are required for the development of B cells in the bone marrow. Thank you.